Okay, can everybody hear me all right and see my slides? Awesome. Um, so thank you so much, Mike, for having me. I can't express to you how excited I am to be here. As Mike mentioned, am an alumna of the CSULB biology program, and I actually did my studies in Salmalek. So I did start out as a marine biologist, and then I kind of switched over, but I'll get to that in a second. So I'm here today to tell you about my research that I've been conducting on the origin of life and what an understanding about the origin of life can tell us about the potential for life on other worlds. But before I do that, I do just want to take a second and briefly acknowledge my history with CSULB and the program that kind of started me off on my academic journey, because I don't really get to talk about that very much, and I'm hoping that that will be informative for you. And then from there, I'm going to just give you a very brief overview of the field of study that I'm currently in, called astrobiology, because as a relatively up and coming field, I realize not all of you may be familiar with astrobiology. And somewhat selfishly, I'm super excited about that because it means that I get to tell you about it. And I can just remember, being an undergrad, sitting, well, not in the virtual room, but in the actual room in seminar. And I just keep imagining if someone had told me about astrobiology, maybe my path would have been a little bit different, or maybe not. So anyway, I'll just give you a brief whirlwind tour of that just to put my research into kind of a larger context. And then of course, I'll get into my research. So let's get started. Oop, see if I can advance. Okay, so when I knew I was going to give this seminar, it automatically sent me back down memory lane, and I quickly went through my pictures and tried to find kind of photographic evidence of my time at CSULB. And I did find some. So what you're seeing down on the left-hand side is my first ever gel that I ran. And I was so excited about it at this time that I took a picture. And if you notice, there are actually two gels. There's like a tiny one. That was the one I loaded because I wasn't trusted with the big gel. So anyway, I just thought that was fun. And then in the middle is a picture I took of some peanut butter and jelly medium for Drosophila larvae that I actually took in what was it, laboratory for cell and molecular biology, what was biology 440L at the time. And this was Dr. Eldon's module where we learned all about flies and developmental biology. So again, like those memories are so fresh in my mind and I love it. And then the last picture on the right is me during my training as part of the Bridges stem cell program, which I did in my last year at CSULB. So as part of the 2014-2015 cohort of CERM stem cell trainees. And so I spent that year doing research at City of Hope on the role of epigenetics on cancer cell drug resistance. So I got kind of a very broad training, both in coursework and in research in the biological sciences. And I focused primarily on molecular biology, although that in and of itself wasn't a linear path. So as you heard, I started in marine biology and then kind of my interest evolved and I decided I was more interested in the molecular side of things. But then I guess the reason I'm bringing this up is because, as will become very apparent in my talk, hopefully, is that I've come quite a long way. So in terms of my research interests and what I'm studying now, things are very, very different. And so what I'm hoping that you'll get out of this presentation, if nothing else, is that interests change. And that's part of the beauty of being a scientist and being a human, really, is that sometimes what you find interesting and what gets you excited changes. And if you have the opportunity to capitalize on that and to take new opportunities that allow you to pursue those interests, if it makes sense to you, you should. Even if it's really scary and even if it means maybe leaving behind some of the things that you thought you were an expert in to become an expert in something else. So I'm happy to talk about my academic journey more than I'm able to kind of share right now. But I hope, like I said, if nothing else, not to get all philosophical or anything, but follow your interests because sometimes they'll take you really crazy places, including working for NASA, for example. So anyway, given my background and that you now know that my interests have shifted wildly in my academic career, I kind of want to shed some light on the thing that really was transformative for me, and that's astrobiology. And like I said, I'm really excited to be able to tell you about it, and hopefully you'll see kind of why I made the change that I did and why I haven't looked back. So what is astrobiology? So astrobiology is a field of science that is defined as the study of the origins, evolution, particularly the early evolution, distribution, and future of life in the universe. And this is a kind of formal definition that's advanced by the NASA Astrobiology Institute, which is the biggest funder and organizer of astrobiology research, at least in the US. And as you might be able to guess from the huge breadth of this definition, is that astrobiologists come in many flavors, right? So it's a highly interdisciplinary field. You have biologists, you have chemists, you have astronomers, geologists, anything under the sun. And that's because it's such a complex question that requires understanding from so many different areas of science that really, to be successful, it requires coordination and collaboration between lots of different kinds of thinkers. And this image here, I hope, kind of captures that. Although I will mention that one thing that's not reflected in this little graphic here is the humanities. So the humanities also play a huge role in shaping the direction of astrobiology. As you might imagine, when you're looking for life on other worlds, things like ethics and philosophy come into play. So hugely interdisciplinary field made up of scientists who themselves have interdisciplinary backgrounds, and I count myself in that category. So really, really cool stuff. But now I just want to step back and, one of the things that makes astrobiology great is the kind of interdisciplinarity, but another thing that makes astrobiology great is the kinds of questions that it allows you to ask. So questions like, what is life? Like, really, what is it? What makes it so special? What makes it different from non-living things? How did life get started on this planet? What happened? Why did it happen? What conditions were necessary? Could it have arisen on other worlds? Does it exist currently on other worlds? And if it does, how do we detect it? Where do we look? What kinds of instruments do we send? What measurements do we take? And finally, what is the future of life in space, right? As we continue to expand outward and we discover more and more habitable worlds, what does that mean for the future of Earth life? So, I mean, again, I'm an astrobiologist now, and it still blows my mind that these are the kinds of questions that we get to ask as scientists. I mean, these are questions you've asked yourself probably, right? At least once in your life, if not multiple times. And I just think it's so, so cool that you can transform this kind of deep existential need to understand why you're here, where you came from, into an actual scientific career. And it's just mind-blowing to me. And when I learned that this was a real, legit area of science where you could get funding from NASA on top of that, I was sold, and I've never looked back since. Okay, so with those questions in mind, I do want to take another giant step back and kind of draw your attention to a big problem in astrobiology, but also for me, the reason why it's so exciting to be an astrobiologist now. And that's the two biggest discoveries in astrobiology have not yet been made, okay? And the two that come to mind are the fact that we only have one example of life to date. We have not found any conclusive evidence of life on other worlds. There's some question about whether there are other forms of life that have been created on this planet, either in silico in a computer or using synthetic biology approaches. But we do know for sure that up until today, we have no evidence of life on another planet. And the second big, big mystery is the origin of life. We are clueless, and I tell you, clueless about the origin of life. We have no idea what happened or why it happened. And so, for some people that might make them question, well, what can science tell us about this thing? If we've been trying for so long and we still are clueless with respect to these two questions, does that mean that maybe we will never be in a position to answer that? And that's a valid point. But the good news is that at least I can tell you that current and aspiring astrobiologists are strong optimists. I think that's kind of a prerequisite to be an astrobiologist, is that you have some degree of optimism about our ability to answer those questions. Otherwise, it might be a little bit difficult. But personally, I'm optimistic that we are not only going to make those discoveries, but are going to make them very soon. And for you, who might be discovering astrobiology just now, as I'm telling you about it, it might mean that if you kind of contribute to the cause, you might be responsible, at least in part, for helping shed light on these really, really deep-seated mysteries. So anyway, that was my way to sell astrobiology to you. And from now, I'm going to kind of focus in a little bit on the specific questions that I'm interested in in my thesis research at UW-Madison, and just kind of focus us in a little bit. But I think that I just wanted to give you this larger context to show you that in addition to what I'm going to share today, there are other flavors of astrobiologists doing very, very different things. And if you're curious about those, please feel free to reach out to me. I'll have contact info at the end. I would love to help you navigate this field and what opportunities might be in store for you. So, okay. So let's dive into my research. So the questions that are guiding my research are really kind of an extension of the problems I just laid out for you. And for me, it's the fact that we really don't understand what life is. Part of the reason we haven't resolved those two big problems of the distribution of life and the origin of life is because we still don't have a good handle on what life is. And that might seem really surprising to you, because I think we all have this intrinsic, even somewhat instinctive ability to tell living things apart from non-living ones, right? So if you look around the room you're in right now, you can kind of point to things and pretty conclusively say, that's alive, that's not alive, right? So you kind of have this ingrained notion of what's alive and what's not. And some of that notion might be shaped by things that you've learned that are characteristic of living systems. So I remember in Bio 211, when I took it, I remember going through a list of different things that were characteristic of life, right? Its cellular organization, the fact that it metabolized, the fact that it had genetics and could transfer information, the fact that it could reproduce, evolve, all these things, okay? So we have a nice list of things that to us are characteristic of life. What we don't know is whether any of those things are required for life to emerge. I hope that makes sense. So in other words, we don't know which of those features are essential to life, and we don't know which ones are just consequences of life's evolution. And that can seem really surprising given how much we know in biology. The fact that we still don't have a clear grasp of life as a general phenomenon. And I think that personally, I think that's what's keeping us from resolving these larger mysteries. And until we can make progress in this area, I think it's hard to imagine resolving those issues. So another problem that's kind of comes out of this lack of understanding about life as a general phenomenon is that we don't have a consensus definition for life. Okay, so at this point, I would say that there are as many definitions of life as there are people. And that's great, right? That in and of itself says something about what we perceive as life. The problem is scientifically, it just gives us a moving target, right? And if we're trying to study a phenomenon without actually knowing what the phenomenon is, that's particularly difficult. So in astrobiology, we resolve that problem the way you would in other fields, right? So there are analogous problems in other fields. So in evolutionary biology, I like to compare it to the species concept problem. So the fact that there's no like one clear definition of a species. So we do the same thing. All we do is we just clearly define what we mean by life. So that's what I'm about to do right now. So the definition of life that we kind of tend to be partial to in my lab is that life is a self-sustained system capable of undergoing Darwinian evolution. Now, this was a definition that was the result of a workshop that NASA put together almost 2 decades ago now. And they basically were tasked with trying to come up with a consensus definition. And it turns out it was really hard, but this is the one that they kind of honed in on. And as you can see, it really features two things, right? So the two things that it highlights are this ability for life to sustain itself. So this self-sustained kind of makes you think of things like reproduction, self-replication, right? And then the other aspect is this ability to evolve, right? And specifically to generate complexity through evolution. So these are the kind of two processes that most people, I would say, in the astrobiology community are now recognizing as the two likely candidates for essential features of life. So there we go. So that's our definition. And so really then, with that definition in mind, the origin of life problem can basically be rephrased as the origin of those two processes. How did self-propagation, self-sustenance, and evolution arise? What were the first entities on Earth to be able to display those two properties? So that's kind of the question guiding most origin of life research. And there are different camps, okay? And I'm partial to one camp, and you'll soon see which one. But I'm going to present to you a few of those camps just so that you have a pretty good handle on kind of the state of origin of life research these days. So we have compartment first camps who tend to view the fact that life tends to be organized in these little bags of chemicals as kind of the defining feature. And so they're working really hard to explain how maybe simple bags of chemicals might emerge on the early Earth as kind of a model for how life might have gotten started. Then you have more of a genetics first camp that tends to view life's characteristic feature of being information based and storing information in these linear polymers. And so they're working to try to figure out how those polymers could assemble out of conditions on the early Earth. And then finally, we have this metabolism first view, which favors the networks of metabolic reactions that compose life, and trying to argue that that was the first living entity capable of displaying these processes. So of course there are other frameworks for the origin of life that kind of fall in between them. And I would say at this point that the one that has the most traction in the astrobiology community is metabolism first. And the reason being that if you think about it, and hopefully this will become more obvious as I continue, metabolism can actually capture some of the features of a genetics first and a compartments first model. And I'll get into that a little bit more, but just so you know, metabolism first seems to be winning so far. Okay, so with that in mind, now I do want to take an opportunity and tell you about something that we do know quite well in the origin of life community. And that's that the building blocks you need to make those structures I just showed you are actually pretty easy to make. And that's interesting because for a long time, origin of life science tended to think that if you could resolve this problem, you'd resolved the crux of the origin of life problem. So if you could figure out where the building blocks came from, you'd solve the origin of life problem. Of course, we know that that's not true. And since then, we've also figured out that this isn't really a problem at all. Turns out you can make life's building blocks really easily in the absence of life. And one is through spark discharge chemistry. Now, I remember learning about this in Bio 211 and being blown away. But it turns out that scientists a long time ago, now almost 50 years ago, discovered that if you mixed a bunch of simple gases together, like water vapor, methane, CO2, nothing sophisticated, and you ran an electric discharge through it, you could make a bunch of life's building blocks, including amino acids, nucleobases sometimes. And so this was a really seminal experiment that basically just blew everything open and made people realize that, oh, so the building blocks can be made very easily, in fact, and the early Earth was probably just chock full of it. And so here's just a little picture of a setup for that Miller-Urey experiment. You can see it's relatively simple. And since then, we've also found lots of other ways you can make life's building blocks. So hydrothermal vents, which are these kind of chimney systems at the bottom of the ocean where ocean water interacts with a newly forming crust and releases a bunch of nutrient-rich water in these chimney systems. Those are basically big factories for this stuff. And finally, maybe most elegantly, we've also discovered that the organics needed for life are readily made in space and are being flung at us from space. In fact, we have evidence that the building blocks of life, some of them, might have been formed at the same time as our planet. So in the early days of the solar system when it was forming, we also had all these organics forming. And then they were eventually delivered to Earth through the accretion process. And so that's maybe where we have our origins. So our building blocks might have cosmic origins in and of themselves, which I just think is crazy. So all that to say, to give you examples and show you that the building blocks are not a problem. Turns out the Earth was probably just littered with them and they were made by various pathways. Now, the biggest problem, now that we know that, is how do you start from those building blocks and get them to self-organize into a cell, right? Look at the difference in complexity in those two. How do you get from one to the other? That is the major problem, and that's the thing we have not resolved at all. We have some good ideas, but this is still a hugely open question. So this is it. And now I'm going to walk through those scenarios I showed you, the compartment first, genetics first, metabolism first, show you why so far they failed, and then tell you about our model and why we think that it addresses the problems that the other ones can't. So we have the protocells, genetics, metabolism, and the one that I'm going to present to you that we've developed in our lab is this idea of chemical ecosystems. Okay, so what's the problem with these other models? So the compartment first model, like I said, basically is inspired by the fact that life is organized in these complex bags of molecules, and that those bags actually play a really important function, right? They keep the whole intact, right? They allow separation between the interior environment and the exterior. They allow chemicals to kind of stick together and not diffuse away. So working backwards from that complexity, scientists have imagined that there must have been a stage in which cells were much, much simpler, right? So a much simpler bag of chemicals. And this is what we refer to as protocells. So protocells are basically just progenitor cells. They're still bags, but they're maybe not as sophisticated. Now, the problem is that from a logical standpoint, it's still really hard to imagine how even a simple bag of cells that's functional, that can do the processes of life, namely self-propagate and evolve, is really hard to conceive. And in fact, when you do this in a lab, it requires quite a bit of finessing, right? You need to be a pretty talented chemist or biologist to get this to work. So how would this work on an early Earth environment? Knowing that to get a cell to function and keep going and divide and evolve, you would need the bag to somehow miraculously encapsulate a minimal number of metabolites that could sustain themselves, that would feed on food and release waste to grow. So think about everything that's needed for that. You'd need some kind of coordination between the interior of the bag itself. The bag would need to be just permeable enough to let food in, to let waste out. And it would need to persist long enough to be able to divide and create two functional cells. So really improbable. And so far, no one's been able to make a spontaneously forming, dividing, evolving cell in the laboratory. So what does that say about the likelihood of this emerging under prebiotic conditions? So those are the problems with compartment first. An alternative that's maybe easier to imagine than a kind of a fully functional bag of compounds is the genetics first, which presupposes what's often called an RNA world, and you may have heard that term before, which alludes to a part of life's distant past that was dominated by one molecule called RNA. And so people think that maybe this RNA world might have consisted of naked RNA molecules that could display the properties of life. And the reason why they think this is because it turns out that RNA is kind of special because it can play two big key functions that are important to those processes that we're after. One is the ability to store information, and the other is to catalyze reactions as part of metabolism. And ribozymes can do that. So ribozymes are catalytic RNAs that not only store information, but can do things like catalyze their own formation. And so people think that this might represent a plausible first step for the origin of life. And so they've come up with a bunch of scenarios to explain how you could get the precursors of RNAs to accumulate and polymerize and all that. The punchline is no one's succeeded, really. And all of the progress that's been made, again, all has required the talents of lots of chemists and biologists working together. And even with all that effort, we still haven't found an example of a spontaneously forming ribozyme that can catalyze its own formation. And there are a bunch of other problems too, but I think this is kind of the one that exemplifies the biggest problem, is that it's just not plausible to imagine the origins of a self-replicating catalytic RNA spontaneously forming by itself in the absence of a prior living process. So that's the problem with the RNA world stuff. And then, in my opinion, the metabolism first one is the most promising, but there are still some flaws too. So metabolism first basically just looks at the complexity of modern metabolism, which is just the net sum of all of the reactions that take place inside a cell to maintain the interior environment. And imagine that maybe life started as simpler metabolic systems. So there are some people who are working to kind of jumpstart analogs of metabolism without using any kind of sophisticated enzymes, and just purely starting from the raw materials of the early Earth, and to some success. So some people have shown that you can actually jumpstart what's known as the reductive TCA cycle or the reverse TCA cycle only using minerals, which is super, super cool. It suggests that our metabolism might be the way it is because it was favored on the early Earth, which I think is really cool and elegant to think about. But the problem is, it still doesn't get us to that question we're trying to answer, is how do you get a fully independently propagating and evolving system from one simple metabolic network like this? And the piece that we're missing is this notion of autocatalysis. So autocatalysis is just a fancy name to refer to a set of chemical reactions that self-amplify, okay? And actually, this is the basis for all of metabolism. Metabolism is self-regenerating. It makes more of itself. That's its primary function. And if you boil it down to its simplest components, you actually realize that autocatalysis is really simple. And it turns out autocatalysis is pretty much inevitable. You see it everywhere. If you mix enough compounds together, it doesn't matter what they are, you will get autocatalysis. So what is autocatalysis? So here's an example. It's just a very simple example here, 4-membered autocatalytic cycle. So we have a molecule A that can help catalyze the formation of a molecule B, which can make C, which can make D, and then here's the key. D can help make 2 of A. So you can see by running this cycle, what happens is that you make more of A, and the whole system grows exponentially, as long as you have a constant influx of food. And food molecules here are denoted as a little letters. So little A, B, C, D, those are food that might be present on the environment. So the idea is if you have these food compounds and you happen to make 1 of A, that's all you need to get this cycle, this autocatalytic cycle of amplifying molecules started. And so that's cool because it suggests that we actually have a candidate for a system that approximates properties of life that we value in our definitions today, that's simple enough that we don't have to implicate any kind of talented chemist to get started. The problem is, like I said, we don't really know how evolvability takes hold. And so there's been lots of research, mostly theoretical, to try to explain how that can happen. And one is that you can imagine that these cycles, after they establish, might change over time, right? Maybe some new reactions get added on, and that's what's depicted here in different colors. They're called reaction cores. And if we go back to our other diagram, you can see what might happen. So what's not drawn in that original figure is that for each reaction, there's waste products being generated. And mostly, those waste products don't interact with the cycle. They just diffuse off and get lost in the environment. But maybe on occasion, one of the waste products can react and form a new compound, E, and that sets off a new cycle that's dependent on food on the environment. And so you can see how you went from a 4-membered cycle to an 8-membered cycle, but that 8-member cycle could only appear if the 4-membered one had appeared. So that's kind of this core model of autocatalysis and how it might evolve. So in our lab, we've been working to expand this model and try to put it like in real terms, right? Because what I've just shown you is just abstract. It's just theoretical, it's just simulations. What if we actually imagine these things taking hold? What would we need in the environment for them to get started? What would we need to see them evolve? What is it? Like how, because I'm an empiricist, I'm not a theoretician. So I need to see things happen rather than just imagine them or run them in a simulation. So we've been developing kind of an extension of these autocatalytic models. And what we've discovered is that autocatalytic systems, cycles, behave oddly like biological species. And there's some suggestion that there's continuity between the rules that govern ecological interactions between fully fledged organisms and the interactions between simple chemical cycles as we might imagine them at the origin of life. So to walk you through this model, I've kind of drawn an autocatalytic cycle slightly differently, but it's basically the same thing. So here in this case, we have M molecules that are the equivalent of the capital letter A, B, C, Ds. And we have food that's only present on the reactant side and waste only present on the product side. And here, this little Pac-Man diagram is what I'm going to use in the rest of the talk. But you can see how M, which we call our member species, is the autocatalytic species. It's the one that's generated more as a result of this cycle. And you can see the food coming in and the waste coming out. So what we found when we modeled the growth behavior of these, even the simplest autocatalytic cycles, is that they behave like biological populations and that we can describe their growth using logistic growth models, which is exactly what we do with real biological populations. So they display a carrying capacity beyond which the population collapses. They have an intrinsic rate of growth. And so this was really remarkable to think that we could approximate the behavior of these simple chemical systems using rules we normally use to describe fully evolved biological populations. And even more interestingly, when we implicated multiple autocatalytic cycles that interacted in some way, either as competition, so we made them compete for food that was present in the environment, or as mutualists, so one would generate a waste product that could be consumed by the next, or as predators, we had whole cycles consume the other cycle, is we found the same thing. They behaved oddly like biological populations. And if you're taking ecology at all, you might recognize this pattern here. This is typically described by what are called Lotka-Volterra models, so predator-prey type dynamics. And the same thing happens in these chemical systems, which is really quite remarkable. So again, it just suggests that there's this continuity between the way full-fledged biological populations behave today and how these autocatalytic cycles behave, which is super cool. So one of the main takeaways, or one of the main suggestions out of these models, and something we're working on now, is to explain how those populations might evolve. And so we're working on modeling increasingly complex systems with more than one autocatalytic cycle, lots of different kinds of food. And we think what we might see is that evolution becomes a consequence of different ecological interactions between these populations. And so far, we have some interesting results. So succession might be another word you recognize from ecology. We see that too. And that's a phenomenon where the presence of one cycle actually primes the environment for other cycles to take hold. So really fascinating stuff. And we're really hopeful that this model is going to tell us even more about how these ecosystems, chemical ecosystems, might represent a path to complex cellular life. Now, another element I need to bring in at this point, now that I hopefully have convinced you that these chemical ecosystems, despite how simple they are, can actually teach us a lot about these fundamental principles of life, are how you can actually imagine these cycles emerging. Because if you imagine chemicals just mixed in, say, an ocean, which is a likely scenario for where the origin of life took place, what would happen is you'd have this dilute mixture of stuff, and it wouldn't be very likely for those molecules to ever come into contact and create these cycles. So one thing that's been proposed is the role of mineral surfaces. I also remember learning about this in Bio 211. So it turns out that minerals are just big solid chemical structures, and because of that, they can actually stick to compounds, including many of the organics that are important to life today. And so interestingly, back in the late 80s, a German patent lawyer, of all things, proposed that minerals, specifically pyrite, which is shown here, which is an iron sulfide mineral, could host little microcosms of these autocatalytic cycles and host them long enough and protect them so that they could eventually give rise to life. And that's an idea that's very popular in the origin of life. And there's some version of minerals at the origin of life, whether it's hosting these little microcosms, or catalyzing a reaction. Minerals are very popular in astrobiology. And another one that's often implicated is Montmorillonite Clay, which I also remember learning about in 211. See, I learned a lot in 211 about the field I eventually became a part of. But the cool thing about Montmorillonite is that it has this kind of like sandwich structure. And those sandwiched layers can actually hold on to different kinds of molecules and catalyze reactions between them. So there's been quite a lot of research to show how Montmorillonite clay can actually act almost as a protocell and facilitate these reactions that nowadays take place inside a cell. Okay, so not sure how much time I'm going to spend here because I'm kind of babbling a lot, but I just wanted to convince you even more of the fact that these autocatalytic cycles can evolve, because I know that's often the thing that people get hung up on is, that's great, Lena, but there's no genetics here. So how could you possibly have evolution? And that's a really good point. And the way that I will respond to that is that we think that these systems actually harbor information the same way that genetic polymers do, but just much better, right? So genetic polymers, we think, are just an optimized version of what these autocatalytic cycles do already. And that's if you allow yourself to imagine that each of these cycles contains information in their composition, right? So as the cycle churns out, it will maintain that composition. It'll just expand it by the number of times that cycle is increased. So in a way, it's maintaining a state. And if that state changes, right, if you now have the addition of a new core, the way I showed you in this example, you've now changed that state. You've essentially mutated. And so we imagine that this kind of analog type information might have been a stepping stone to the addition of genetic information later on. And so just to help you imagine, look at the next slides. This here, these kind of colored dots, these are just the same representation as this, just kind of simplified. And so we've developed other models in our lab to show how these autocatalytic cycles that have a defined composition here that we can imagine on an abstract space defined by chemical composition axes, how those might change in chemical composition depending on what changes happen. So you can see here there's a yellow change, and then there's a blue change. And if we imagine what happens in real space, right, since we're talking about microcosms on mineral surfaces, we could imagine kind of these slimes coating the mineral surfaces and changing in composition as a result of these compositional mutations. And those are free to explore their own trajectories. And eventually, because you have an active environment that's imposing selection, right? So as these mineral surfaces are being continually exposed and drawn down, what that's effectively doing is selecting for variants that are able to get from grain to grain. And so what's shown in this figure here is that the red variant, the red trajectory, gave us a colonizer that was better able to colonize neighboring surfaces, and it essentially won out. And so we call these systems, these kind of absorbed autocatalytic cycle slimes, for surface limited molecular ecosystems. So that's what I'll call them for the rest of the talk. And this is also just to say that we've also come up with potential paths to show how selection for increased mineral colonizing ability could represent a plausible pathway to cells, so to C-cell stages that can persist away from the mineral surface. And I'm happy to point you in that paper if you want more. Okay, so last part of my talk now is going to be just focusing on the part I've been focusing on in my PhD research. So theory is great. Everything I've just shown you, I find fascinating, but I'm an empiricist, right? I need to see this stuff happen in a lab to believe it. And so that's exactly what I've been trying to do. So I have funding from NASA to try to turn this ecology framework into something we can deploy in a laboratory and study these slimes. If this is a valid model for the origin of life, we should be able to do it pretty easily in the lab. And so to do that, we've developed this approach that we call chemical ecosystem selection. And if you've heard of experimental evolution on things like microbes, it's basically just like that, except in this case, we're deploying it on chemical systems, not biological systems, and we're selecting for the ability to evolve. So what we do is we basically take mixtures of food, right? So things like building blocks that we think might accumulate on the early Earth. We incubate them with mineral surfaces, give some time for these slimes to establish themselves, and then we transfer a small subset of the grains that have been hopefully colonized into a new vessel containing fresh food and fresh mineral surface. And we do that over and over again so that by the end of it, we've selectively enriched for systems that were better at getting from grain to grain. And in this case, it's the yellow, right? So we're essentially imposing natural selection. So you have to be able to propagate fast enough to make it to the next generation. So that's the principle behind chemical ecosystem selection. And the cool thing about it is that it's chemically agnostic. What does that mean? It means that it doesn't presuppose any specific chemistry. And for the origin of life, that's really useful because, like I said, we have no idea what conditions are necessary for these systems to emerge. So this is great because it allows us to input any set of condition that we want. The only requirement is that we have to have a few proxy traits that are related to the rate of propagation of these slimes that we can observe across transfers or across generations to look for evidence for a response to selection, okay? So this is just hypothetical data here, just to show you that we have some notion of what that should look like. So if we keep doing our transfer process, like in the vials I just showed you, and we don't see any change over time, that suggests that nothing has arisen that seems to be responding to the selection. Or alternatively, it can mean that the proxy trait isn't tuned to detect it. But if we see a change, an increase in the rate of propagation over time, that suggests that the serial transfer protocol has selectively enriched for systems that are better self-propagators. Alternatively, we could get kind of more stepwise evolution, where we see a sharp transition that's maintained for a few generations until it happens again. So that's the principle. We're just looking for changes in response to that selection pressure. And so to better illustrate that, I'm going to give you one example of a recipe of one set of conditions we've been trying. We've tried lots in our lab, but I'm going to highlight one that's given us some pretty interesting results recently. So the soup that we used was one that's inspired by those spark discharge experiments that I showed you. So we just basically went in the literature, kind of tabulated what they found, and then just mixed those together in some seawater. And we made a couple additions too. So you might notice that ATP is hiding in there. ATP is not made in Miller Urey. It's a very complex molecule, even though it's pervasive in life today. But I'm going to draw your attention to that because it's the basis for one of the detection strategies that we're using. The mineral we're using is pyrite, that one that Vectors Hauser, 20 years, more than 20 years ago, a long time ago, proposed as a plausible mineral surface that we grind up into powder. And then all of these experiments were carried out under anoxic conditions, so no oxygen, and this is to be representative of the early Earth environment. And so what we found when we did this experiment is systematic changes in the proxy traits that we were measuring. So here what you're seeing is the concentration of free inorganic phosphate that was pulled off that ATP molecule. And we chose this measurement because we thought it would give us a good approximation of energy usage in our experiments. And then on the x-axis is just replicates. So the gray bars are 10 lineages that were independently propagated. And then we measured the amount of free phosphate at the end of these key generations, 6, 12, and 18. And the black bar is the average of 10 controls, which only had one transfer in their history. So this is to help us isolate the effect of those transfers. And what we saw is as we kept selecting, we saw a decrease in the amount of free phosphate. And we don't know what it means. We don't know where the phosphate's going, but all we care about is that these differences seem to accumulate as a result of the selection process. And then the other thing we noticed is a similar thing with UV vis absorbance. So UV vis absorbance is just a way to characterize how much light is being absorbed by the liquid in your sample as a proxy for how many organics and light absorbing compounds are there. And what we saw is as we kept selecting, same thing, less and less light absorbing compounds, including some of the organics in our solution. Where are they going? We don't know. But these results were kind of tantalizing enough that we decided to repeat the experiment carried out for much longer, and this time measure at every generation. And this is what we found when we did. So each of these lines are each of the independent lineages, and same thing, amount of free phosphate at the end of a generation. And what you see pretty clearly is that all 10 of those lineages oscillated. And that was really, really cool. So that initial decrease, for most of the lineages repeated. But then what we saw at the end of generation 10 is a rapid reversal in direction, only to have the pattern repeat again. Now, there are lots of ways to interpret this oscillatory pattern, but it is consistent with the appearance of autocatalytic cycles. And the reason being, oscillations tell you there's some kind of nonlinear feedback in your reaction. You can't get oscillations without feedback. So at first glance, it seems consistent with our hypotheses. And then this here is the same data just averaged out over the 10. And you can see that clear oscillatory pattern. And then those red lines are controls, so they did not oscillate. Oscillations only appeared in response to these transfers. And interestingly, when we compared to our experimental results, they actually line up with that predator-prey scenario, which again is just kind of maybe making false equivalencies, but is tantalizing and suggests that there may be interacting autocatalytic cycles appearing. And then just a quick little cool thing also, when we looked at the grains of samples that had lots of transfers, we saw these really neat fractal structures that appeared on the grains that weren't present in controls. And we're still trying to make sense of what those mean, but at first glance, it's consistent with stuff being stuck onto the mineral surfaces. So all these things are consistent with our notion of what we should see if evolvable autocatalysis has appeared in our reactions. But of course, much more work is needed. And right now we're working on extending those experiments to much further out, so at least 100 generations. So we can try to see if we detect a response to selection. And we're also refining our theoretical models to include the presence of mineral surface, which we haven't done yet, to just help us better align the results of both of these approaches. And so here are my conclusions. I know I'm at about time, but just to wrap up, just to say that, you know, this chemical ecology framework for me has a lot of promise. It helps resolve a lot of the problems that other conventional approaches fail to capture. And as a bonus, we have a clear path towards experimentally validating this, which we don't get in the other scenarios. And as another bonus, we've already landed on at least one set of conditions that yields promising results. And so we're further investigating those. And then the last thing I'll mention is we're also working closely with groups who are working on upcoming missions to Titan and Mars to see if we can recreate conditions representative of those environments and see if those conditions are conducive to these similar types of processes. So in a way, it might help us also answer this question of the potential for life on other worlds. And with that, I'll just acknowledge my lab, especially my PI, David Baum, our collaborators, and you for your attention.
I have a question. Would you be able to explain again why you added ATP to that like soup mix that you were doing at the end? Because I feel like you mentioned that it's a very like complex molecule that I don't think would just be like out there, you know?
That's a great question. So when we were designing the soup, we wanted to include potential sources of energy. And when we first designed this soup, we were just trying to design something that would maximize the likelihood that we would get at least an interesting result so that we could further validate the approach itself. And so in our first pass, we were trying to be true to what we thought the early Earth environment would host, but we were more interested in getting kind of a positive result. And given our knowledge of how pervasive ATP is as an energy currency in life today, and its relative stability compared to other forms of phosphate, we chose to include it anyway, knowing that if we got cool results, we could swap it out potentially with another more plausible source. So yeah, so that's my quick answer. There are other reasons too why we picked it. It turns out phosphate's a really difficult molecule to work with just because it's so insoluble. And that's actually a big problem. There's a whole thing, the phosphate problem of the origin of life. If you Google that, like, yeah, phosphate's a big problem.
Cool. Thank you very much. Appreciate it.
No problem.
How much time elapsed between generations? I can't remember if you mentioned that, so that we have an idea of the time scale that you were looking at.
Yeah, I did not mention it. In this experiment, it was 2 days, and that was purely arbitrary. So we have no theory to help us determine the right generation time. It was kind of the luck of the draw. We went with what was convenient. And we were worried about that because if we didn't wait long enough, right? So if we transferred maybe at a day, it's possible that we wouldn't have given enough time for these slimes to establish themselves. And if we waited for too long, the systems might have reached equilibrium. And at that point, we weren't selecting for anything. We were just transferring dead stuff. So we went with 2 days and decided we'll see what happens. And we have been doing experiments now where we play around with that generation time, and it does seem to matter. How it matters exactly, we're still figuring that out. But like, if you modulate that, you get very different responses, which is really intriguing. But yeah, so 2 days in this experiment. Oh, should I go in the chat maybe? See if there's any questions.
I've not seen any questions that we've missed in the chat, just a whole lot of enthusiasm about the presentation and your enthusiasm for the work, which I would echo. I mean, this is really, really neat stuff.
Cool, great, I'm glad to hear that.
I had a question for you in terms of the initial definition of life. If you're saying that it's self-sustaining and capable of responding through Darwinian evolution, there are a number of accounts where a piece of software can do that.
Yes,
which is really cool. When I am instructing in my class, one of the hallmarks for life that I like to look at is the idea that life is homeostatic, in that it is separate from the outside environment and keeps itself protected from the outside environment. And you can understand a lot about energy and metabolism and immunology and membrane properties with the central idea that all of these things are mechanisms by which a homeostatic living organism protects itself from the insults of a variation in the outside world through homeostasis. How does homeostasis fit within your ideas of life in these models?
That's a great point. So I guess the slight pushback I would have to including homeostasis as it's traditionally defined, I guess, in biology, is just how, so we have this notion that life is homeostatic because it has this cellular structure. And even when you look at, multicellular organisms, there's this idea of self, right? That's very clearly separate from its outside environment. But really, chemically speaking, life is an open system, right? So it's constantly exchanging with the environment, but in a regulated way, right? So in a way, the question of homeostasis is at what point, what is that? Did that change, right? Was it a vastly open system at its origins and the product of evolution is it basically getting more and more closed off, more and more regulated. But I do think this maintenance of self and separation with the environment, being able to regulate independently of the environment, is a hallmark of life. And I would argue that it's kind of embedded in that idea of self-sustained, because really the requirement for self-sustenance is being able to maintain disequilibrium with your environment. And that's kind of a fancy way of just saying that you maintain a state that would otherwise go to entropy, right? It would kind of break apart if energy wasn't being supplied to maintain it. So I think the idea of homeostasis is super important. It's just what form that takes and how important that was and what form that took at the origin of life. That's a huge question. I mean, I hear some people argue that, you know, hurricanes are homeostatic in some respect, but I think most of us would agree hurricanes aren't homeostatic, or they aren't alive, and they're not as homeostatic as living systems. So yeah, really, really interesting. And I think your point just gets at this problem of how do you capture all of that in one definition?
Awesome, thank you.
I have one more question, because I was really caught on to that idea of how you can make the building blocks with static shock. Has that been found anywhere else? Because in like any other place in the universe or like in our solar system.
You mean specifically fed off of like lightning bolts?
Oh yeah, or any of that.
Yes. So prebiotics, and we call this general process of making life's building blocks without kind of with it in abiotic settings, prebiotic, because we use the term prebiotic because it like comes before life, but also because it's related to life. We find evidence of prebiotic synthesis almost everywhere. So for example, Titan, the reason why it's so much of interest to NASA and why we're sending a mission there in about a decade is because we have evidence that it's just loaded with biologically relevant molecules. And in that planetary system, it's made through interactions between this really thick haze that Titan has and the strong UV that's being shed off of Saturn and Jupiter. And so you have a similar type of process. The energy source is different, but it's the same kind of thing, just taking simple building blocks, feeding an energy source, and then getting more complex stuff out of it. So yes, to answer your question, it's pretty much everywhere. There's so many different ways to make prebiotic building blocks, which is fascinating in and of itself. And just begs the question, why do those building blocks sometimes make life and why other times don't they?
Yeah, if they're so widespread, then it must be like really insurmountable processes to just combine them all together.
Maybe, yeah. Or life is pervasive and we're just not looking for the right things.
Yeah.
We're tuned to our definition, and so far all of our detection methods are calibrated towards those metrics. So and if there was some really exotic form of life that ultimately did those processes that we value in our definitions, but were made of entirely different types of molecules, for example, we would be totally remiss. Like we wouldn't see them. So, which is also, that keeps me up at night all the time. Like
that's so scary. I was watching a bunch of horror movies from like the 1980s about like The Thing and all that. So I'm like, wow. Thank you.
Yep.
Why did you end up using the pyrite outside of the earlier reference instead of the Montmorillonite? I can't remember.
Yeah, Montmorillonite. Yeah. So again, we just kind of, we went for crowd favorites, right? So we went in the literature and we're like, what are the minerals that come up all the time? And which ones are easy to work with? Clays are not that easy to work with because they just love to stick to themselves and it's really hard to get them to cooperate. They also can very easily be contaminated because they're big sponges, basically for organics. It means that if we don't like clean them diligently and do a lot of pre-experimental analysis, we could be confounding our results. So we went with pyrite just because it's easier to handle, but because it also has been in the literature for a long time as being of particular importance. Other things that drew us to it, it was very abundant on early Earth. There was a ton of it. And also, it is basically like a mineral form of an iron sulfur cluster. So most enzymes that are conserved across life, their catalytic cores are iron sulfur clusters, which are basically just pyrite. So you can view enzymes like the conserved enzymes as pyrite wrapped in protein. So that was another reason why we were kind of drawn to it.
Michelle had asked in the chat, what does RNA and DNA come into play with your theory?
That's a great question. And that's another area that we're tackling right now, I didn't really mention, is the transition between a purely analog system where you have kind of simple molecules in an autocatalytic cycle, and where digital or linear encoding comes in. So we think that under the right conditions, if you have a autocatalytic cycle kind of feeding a polymer, if you tweak conditions just right, you can start to favor both. And what we think we'll find is that systems that have this linear component, so the way you would have with an RNA, tend to outcompete others. So we think there's some fitness advantage to having a linear component in the sum total of the information that's stored. So we think that probably in our model, right, simply put, we think that the first kind of precursors of life were these autocatalytic cycles, maybe nested on these mineral surfaces. And as they were selected by natural processes, maybe they incorporated a polymer, and that incorporation suddenly allowed the systems to persist better, or maybe colonize more surfaces or what have you. And that could explain the transition from having purely analog information to having both. So that's what we're thinking. And then maybe adding DNA later, that's kind of harder to explain. One of the other big problems that we're not really tackling in our lab is the origin of translation, which from a physical chemistry perspective makes no sense. Like the fact that you have these three interacting polymers, DNA, RNA, protein, thermodynamically is really hard to explain. And so it's kind of makes origin of life scientists scratch their head and go, why the heck? Why? You know, why are there's these two genetic biopolymers? And it's possible it's just a frozen accident, right? It just worked. So there it is. But yeah, so that's kind of where linear polymers fit. And RNA is kind of like the obvious one.
I really appreciated your talk. I thought it's also really cool just to see a Cal State Long Beach alum doing awesome things. So that was really cool. And I think one thing that it's not anything specific to your presentation, but overall, I like that you took some pretty complex ideas and you were able to simplify them down into a way that isn't so jargon heavy and that a lot of people can understand. And I think that just as scientists in general, we need to be cognizant of that and understand that we need to education, and not just for our science colleagues, is really important. So I just want to say thank you for a really great talk.
Thank you. That means a lot to me, because I love getting people excited about this stuff, and there's no better way to do that than to make it understandable. So it means a lot to hear that I've been at least partially successful with that. So thank you. Thank you.
So this might be coming out of absolute left field. Actually, no, I'm going to let Eric ask the question first. Go ahead, Eric.
It's also kind of a left field question. And it's, as an astrobiologist, are you in the camp of the phosphine signatures in Venus being just totally screwed up and they're just over reading the noise, or there actually is detection? Because all the new papers coming out they're arguing one of both sides. Or are you just Venus?
Oh, thank you for asking that. So for those who don't know, in case, I mean, it made a pretty big splash, but I guess almost maybe a month ago now, I don't even know when it happened. A paper came out claiming that they detected phosphine in the atmosphere of Venus using radio telescope technology. And the reason why that was super exciting is because phosphine in the Venusian atmosphere is very unexpected. From what we know about Venus, having such a reduced gas, so having so many hydrogen molecules, is really hard to explain unless there's some process by which phosphine is being continually produced. Now, on Earth, phosphine is associated with biological processes. So we find it in like cesspool tanks, we find it as a byproduct of some microbial metabolism, even though we don't, which to me, this is even more mind-blowing, the discovery on Venus. We don't actually really understand where phosphine comes from on Earth. We just know that it's associated with life. So anyway, it got people really excited because one possible explanation for the presence of phosphine on Venus is that there's some kind of biological process putting it there. It turns out that it may have been a blip in the actual radio spectrum. So some kind of corrected ALMA data, which is the radio telescope that was used to make the measurements, may have messed up basically. And when they reanalyzed the data, the blip that they associated with the presence of phosphine disappeared. So there was all this kind of big splash about, needing to retract the paper and all that. My personal take is I'm a cautious optimist. I want to believe that it's there. I want to believe there was life on Venus. Personally, I love the idea. There's actually researchers at UW-Madison who are studying this very possibility of life in the Venusian clouds. And it actually, I've invited him to give a talk at our lab meeting, and we were all like fairly convinced. So I love Venus. I love the prospect of there being life on Venus. At the very least, what I love about this discovery, even if it turns out to be moot, is that it will hopefully send more money to Venusian missions to figure out whether it's there. So like the biggest way to me to resolve that problem is to just go there and send a probe and try to detect phosphine directly, as opposed to inferring its presence from a spectrum. So that's my take. I think that it's hard to know for sure using the radio telescope data, but if it can generate enthusiasm about going to Venus and exploring the possibility, that makes me very happy.
Okay, so if no one else jumps in with the question, I get to ask my left field question here. I'm going to set it up. So cell theory would suggest that the important thing is what's the smallest unit of life, and cell theory suggests that's the cell. And organelles are not independently alive; they're part of the larger living organism. In your definition and quest for origins of life, first part of this question is, how does scale fit in? So are you looking for small things that are life, or are you looking for things that are alive? So if scale is irrelevant, then an argument can be made for an economy as being alive, or a city as being alive, or as a universe or a solar system as maybe being alive. So that's making things big. Second part of this two-parter, contributing also to the left field, is how does time play into this? Because you're looking for something that meets the criteria for life over observable time in your experiments. So that's not seeing things that might exhibit characteristics of life by the microsecond, but that it gets washed out. Similarly, you're not seeing things that might meet the characteristics of life that exhibit these characteristics over millennia, but don't necessarily present as living over what we as humans consider observable time. So, scale.
Yeah.
Really big stuff and stuff on the outliers of the temporal scale as well. How does that fit?
Yeah. So my answer is probably not going to be super satisfactory, because I think the questions you're asking are exactly the reason why we're having so much trouble, getting a handle on how we want to define life, like how we want to package it and what we value in our definition. And so some people argue, kind of similarly to what you're saying, is that the processes of life aren't good enough when it comes to informing our system for discriminating between things that are alive and things that are not, because it's too unifying, right? So I mentioned hurricanes earlier. Hurricanes self-propagate, right? They maintain a state over time. Yes, they feed on fluxes of energy and heat and all a bunch of stuff that's external to them, but that's not dissimilar to what life does. A flame does the same thing. A tornado does the same thing. Similarly, evolution, depending on how strictly you want to define evolution, based on like hereditary units and descent with variation and selection advantages and stuff like that, some argue that that's even still too unifying, because you can argue that there are analogs of evolution happening in crystal formation, for example. That was one idea that came a long time ago, is that crystals were the first living thing, which sounds out there, but actually it really isn't, because when you think about the processes that unify crystal formation and life, they're actually pretty much exactly the same when you describe them in physical terms. So to answer your question about scale, I think that's a really good point. The things that we're looking for, if I had to kind of, I will echo what you said, they're very small, both in space and in time, right? So these aren't things we can observe necessarily with a microscope, right? They're these little tiny blobs that we wouldn't even really know how to look for using any kind of imaging, direct or indirect. They also happen, like you said, in the lifetime of a PhD, right? That's pretty incredible. So, and the way I will answer your question is say that yes, that there are these scale issues. I'm not too concerned about them, and here's why. We're at a stage in origin of life research where kind of any route we take is going to tell us something. And we need a definition, we need to explore it. And if it teaches us that our definition is too unifying, if it doesn't allow us to discriminate between living things and non-living things at a scale that makes sense, then we can refine our definition and start over. And so at this point, I'm not too concerned about recapitulating the scale or any other feature of life as we know it. I'm interested in seeing if this particular approach of aiming for self-propagation and evolution in these simple chemical systems lends any support to the framework of life we currently have. And if it doesn't, right, if we find that any condition that we mix together, we see evidence of these oscillations and that's really all that comes of it, then we're like, okay, well, maybe that's not the right metric. Maybe we need to focus on complexity. Maybe we need to focus on the ability to maintain a state, right? Like homeostasis. Maybe we need to aim for things that are bigger, that leave an imprint that's visible. So all these different things could play into kind of refinements. But because it's such an open frontier and there's really no good theory to guide kind of these first principle approaches, we have to try something. And this is what we've chosen to try. And I think it's teaching us a lot, even with these kind of early preliminary results. So maybe not a totally satisfactory answer, but I think that the questions you raise are at the crux of what's so difficult about this particular question.
No, that was a great answer. Thank you very much. That's very helpful. Very enlightening.
If we wanted to read up more about this outside of the literature that you had in your presentation that I'm sure several of us were rapidly jotting down, where would you direct us?
Oh, okay. It depends. There's such a huge body of literature that can take you down so many different paths. A good place to start if you want to orient yourself with respect to like different flavors of astrobiology and even different flavors of origin of life research is to go to the NASA Astrobiology Institute website. They have a huge list of resources, seminars that are recorded, kind of news if you want to see kind of like what's most recent. And I think that would be a really good way to kind of like tune your eye to what catches your attention. If you want to read more about the work that I'm doing, I think I kind of listed all the ones that are most relevant. But yeah, the NAI, the NASA Astrobiology Institute is fantastic and is actually how I discovered astrobiology was a thing. So it was the result of a weird combination of words in Google. I actually did as I was writing personal statements. My first time I was thinking of applying to go to PhD school, and I was kind of having like an existential crisis, realizing that I didn't know what I wanted to do. Yeah, it was weird. I was looking at cancer cells in a microscope one day, and a weird series of questions came out until I was like, what is life? And I did a quick Google search, and the NASA Astrobiology Institute came up, and I was like, what? So yeah, that's a great place to start, I think.
I see Dr. Eldon's here, so she needs a few rabbit holes to go down.
Hi, Dr. Eldon.
Hi there, great to see you. I was going to ask, although it may be too far off back in your ancient history. So what were you working on at Northridge? Were you in an astrobiology lab there too?
I was not. So I was in David Bermudez's laboratory. So end of my CERM traineeship is when I was thinking of applying to PhD programs first time and kind of realizing I wasn't sure what exactly I wanted to do. Mm-hmm. Kind of had this shift, this paradigm shift in realizing where my interests were. And I decided that I wanted to do a master's. And at the time, I was thinking like, how could I sell my skills as a biologist to the astrobiology community? I was very much thinking of like what can I contribute? Which in hindsight is kind of an interesting way to look at it, because I wanted to learn. Like that was really what I should have been focusing on. So I decided that the best way to do that would be to hone in my synthetic biology skills. So I applied to the synthetic biology lab. And interestingly, my advisor, although was doing biomedical research, had a long history in astrobiology. So he was in Lynn Margulis's lab, who came up with endosymbiotic theory. And he was close to like all of my astrobiology celebrities. So he was not an astrobiologist, but he was well connected. So that was really cool. So the work I was doing was not directly related to astrobiology, but it definitely gave me a lot of tools and networking and just a lot of experience to take to PhD school. So yeah, that was really, really fun. I really liked that.
Okay, that's cool. Because there's a woman there who does some astrobiology stuff, right? Looks at the permafrost and.
That's right, Rachel McElprine. So she was the other one I was considering. The reason I ended up not going is because I was afraid of theory. So math was never like my strong suit, or at least I didn't think it was, and I was really intimidated by it. So you know, and I think some of her work is very experimental now, but at the time I think it was mostly theoretical, and that I had no confidence in myself at the time. It was like, I'm just going to stick to the stuff I know, which is just doing stuff in the lab. But yeah, she does awesome work. So she looks at microbial communities in Earth's permafrost as an analog for what might be happening on Mars, which is so cool. And yeah.
So does that give you any interesting signatures to look for when you're looking at what's happening in your reactions? Or
yes, definitely, because so one of my personal goals with developing this kind of theory and this accompanying experimental approach is that it might become the basis for searching for life on other planets. And the nice thing is there's continuity between how you might detect life and how you might detect like life-like chemistry, especially if you're trying to look for evidence of evolution. So I'm hoping that the signatures that I'm kind of honing in on will help the kind of larger question of how you might detect life in, say, Martian permafrost. But that's still a long ways off, because unfortunately we're not allowed to go to the Martian permafrost. So I only learned this pretty recently, that the caps are actually protected, so you can't go there. The UN came up with these planetary protection rules some years ago that limit where missions from UN countries can go. And the reason is they're afraid of contaminating it, because they think that if you were to get spores there, that permafrost might actually be a pretty sweet environment for them. So although there's so much potential and everyone wants to go there, it's kind of hard to imagine how we can do that safely, at least with the technology we have right now. So that's kind of upsetting, but it makes a lot of sense. I would not want to contaminate Martian permafrost.
cool, thanks.
Yeah.
All right. Well, if there are no other questions, again, I thank you very much, Lena. This was a great and interesting presentation. And icing on the cake is be able to hear the story of a local girl who makes good. And that's just inspirational as far as I'm concerned. So really, thank you very much. I'm going to ask you if you can make me host back again so I can make sure the recording gets archived properly. As we mentioned, I will send you the URL. You can use the recording for anything you want to. If you think of anything that maybe our students at this end might like to see in terms of other links, you can send them to me and I'll put them up on the Beachboard page, but we're at the end of the term now anyway. But anyway, thank you. Thank you so much. This was really just fantastic.
I'm so glad, and this was so fun.